Hello, my name is Soichi Ogata. I am a postdoctoral scientist in Dr. Kencho's lab at Department of Developmental and Cell Biology in University of California, Irvine. Today I will show the dissection and adhesion assay of animal cap explant or organizer explant from frog Xenopus embryo. This procedure can be utilized to study the cell adhesion characteristics and cellular morphology of the cell derived from dissected tissue. This procedure involves in the dissection of a certain tissue from the Xenopus frog embryo and dissociate it in calcium-magnesium-free medium, and then bring that dissociated cell to the calcium-magnesium containing condition on top of the cell adhesion molecule coated slide glass. And we can study the cell behavior, cell migration, and adhesion characteristics of the dissected explant in this procedure. Okay, this whole procedure of dissection of embryo and adhesion assay, I use following special tools. First, this is a calcium-magnesium-free 1x birth 1% agarose plate. And this is just regular 1x birth 1% agarose plate on which you dissect the embryo. And then this is a special dissection tool other than forceps. This is called eyebrow knife, which is a weird name, but this is actually literally made of your eyebrow pulling off from your face and then stick on the tip of pass-through pipette. And the other tool here is called hair loop, which is again your hair making